Section 7.1, Chromosomes and Phenotype. Use loose leaf paper to take notes. Be careful to make any diagrams or pictures that would help you better understand the material. 2 copies of each autosomal gene can affect the phenotype. The traits displayed are going to depend on whether the offspring is inherited either dominant or recessive alleles. The phenotype, the actual expression of the trait, is also affected by which chromosome the gene is located on and whether the gene is on an autosome, meaning chromosomes for humans, chromosomes numbers 1 through 22, or the sex chromosome, which is chromosome pair 23. Everything that Mendel did in his studies were based on traits that were inherited on autosomal genes. Many human genetic disorders are caused by autosomal genes. Because we're on autosomes, the chances of inheriting them are predictable, just like any of these other traits that were studied with Mendel's pea plants. Some of these human genetic disorders could be dominant traits. Some of these could be recessive. Okay, they follow the same patterns as Mendel's pea plants did. Up here you can see these are some human genetic disorders that are caused by an autosomal dominant inheritance pattern, and these traits or these disorders would be autosomal recessive human genetic disorders. If a human genetic disorder was caused by recessive alleles or was inherited on recessive alleles, it would need 2 copies of the recessive allele, one coming from mom, one coming from dad, in order for the offspring to have this disorder. These human genetic disorders often appear in the offspring of parents who were heterozygous. Okay, heterozygous means you would have one dominant, one recessive allele. If this was a recessive genetic disorder, this heterozygous would be healthy. It wouldn't show the trait, but it would carry the trait, so it would be called a carrier of that genetic disorder. It doesn't have the disease, but it carries the gene for it. Since it doesn't have it, it wouldn't show any symptoms, but it could pass the allele on to its offspring. If it was homozygous dominant, it would be completely healthy, not able to pass the allele onto their offspring. And if it was homozygous recessive, this would be an organism that would have this disease, whatever this recessive genetic disorder would be. Here's a Punnett square of a cross between 2 parents that are heterozygous for this recessive genetic disorder. And as you can see in the Punnett square, you're going to get one homozygous dominant, which would be a healthy offspring. Okay, wouldn't have the disease, wouldn't carry the allele for the disease. 2 heterozygous. These would both still appear healthy, but they would both have the possibility of passing this recessive allele onto their offspring. And this would be homozygous recessive. This would be an offspring or that would inherit this recessive genetic disorder. So the phenotypes would be 3 healthy and 1 would have the disorder. Probabilities are 75% healthy and 25% that have the disorder. But with this, you also need to factor in that we have 1 healthy, 2 that are healthy but are carriers, and 1 that has the disease. There are also some human genetic disorders that are caused by dominant alleles. These are much, much less common. Okay, because it's a dominant allele, you wouldn't have a carrier of a dominant disorder, because if you had the dominant allele, you would display this genetic disorder. So again, this is a cross between parents that were heterozygous, meaning both of these would display the disorder. Okay, this one, homozygous dominant, right? These would display disorder. these two heterozygotes would also display disorder, and this one, homozygous recessive, would be a normal offspring where it wouldn't have this dominant genetic disorder. So the phenotypes would be 3 have the disorder, genetic disorder, and 1 would be healthy. By the same line of thinking, the probability of being healthy would be 25%, and the probability of being born with the disease would be 75%. When it comes to genes being expressed on the sex chromosomes, it's a little bit different than autosomal genes because the sex chromosomes, the X chromosome for females and the Y chromosome for males, are much different in terms of what they look like and the traits that they carry. Genes located on the sex chromosomes are called sex-linked genes. So this is a female, XX, this is a male, XY. Genes on the Y chromosome are responsible for development of male offspring, but the X chromosome has more influence over the phenotype of the offspring. The Y is much smaller, as you can see here. Y is much smaller than the X chromosome, and as a result, the Y has much fewer genes, much fewer genetic information to pass on to the offspring. Due to the fact that X and Y chromosomes have different genes located on them, the pattern of expression of these chromosomes is different. Males have only one of each type of sex chromosome. So males would be XY. Okay, this would be male. And females would be XX. Males would display all the alleles on both the X and the Y chromosome. Dominance and recessiveness doesn't come into play at all. There are no second copies of genes, so if you have it, whatever's on the X chromosome is going to be displayed. Whatever on the Y chromosome is going to be displayed in males. An example of a sex-linked, or X-linked in this case, recessive gene would be hemophilia. Okay, so since it's on the X chromosome, it's written a little bit differently. It's the X with this superscript H for hemophilia, or no hemophilia, and X over here, lowercase h, for having this recessive sex-linked trait of hemophilia. Okay, so in this is a male, right? Normal male, right? No hemophilia, being crossed with a female that's a carrier. She doesn't show the disease because she only has one copy of it, but she's a carrier. So if you look at the offspring, we're getting a female, normal. We're getting a male, normal, no hemophilia. We're getting a female carrier, and the male will have hemophilia. So X-linked traits, X-linked recessive traits, are more often expressed in males than females because they only need to get one copy of that trait from mom's X chromosome because the Y chromosome is coming from dad. Now remember in males we said that the males show all the alleles on both the X and the Y chromosome. Females are a little different. Female mammals have an XX genotype. So the expression of sex-linked genes is different from autosomal genes in females. In females, there's this thing called X chromosome inactivation, where what this does is it randomly turns off one of the X chromosomes. It's different in each cell, but by the end of fetal development, about 50% express the X chromosome they inherited from mom. 50% of the cells in the offspring express the X chromosome they inherited from mom, and 50% express the X chromosome inherited from dad. In calico cats, it's kind of easy to see how this is expressed because the female calico cat have two chromosomes with different alleles for fur color. So both alleles are expressed in a random pattern. So you get this black with brown and white. That's because they're coming from different things. Whereas the male just gets one copy of fur color. So in this case, it's just brown. This female, again, different, slightly different pattern. This female, slightly different pattern because of this X chromosome inactivation.